Welcome. The topic of our lecture is genetics and genetic influences in disease states. As you can see, one of our objectives are to describe the structure and the function of the DNA as well as the RNA molecule and have an understanding of chromosomes. We'll talk about how mutations may alter the normal physiologic process of DNA replication, and we're going to spend just a little bit of time on that because you'll do more of that as you write your genetics paper. As well as more in your genetics paper, we'll actually analyze the processes that alter the structure or role of DNA and RNA. We will talk about inheritance and recurrence of genetic diseases, and then use a really nice tool, which is the Punnett square, to determine the probability of transmission in genetic disorders. It's a really cool tool that we'll be able to use. Let's start then by talking about DNA and RNA, because they're critical in understanding both genetics and how mutations might occur. DNA, or deoxyribonucleic acid, is a molecule made up of a nitrogen base, sugar, and phosphate. Now, there are 4 nitrogen bases that make up DNA, and these are adenine, cytosine, guanine, and thymine. Adenine and guanine are purine bases. Cytosine and thymine are pyrimidine bases. Now, this is important because when these marry up, or when these bases pair up, this complementary pairing, a purine base pairs with a pyrimidine base, and they always pair specifically. So adenine, which is a purine base, pairs with thymine, a pyrimidine base. Guanine, which is a purine base, pairs with cytosine, which is a pyrimidine base. And that's a critical piece to remember because that is the concept of base pairing and complementary base pairing. We know that DNA is made up of 2 strands. And these 2 strands of DNA are held together by a hydrogen bond between those nitrogen base pairs. Now, we know that DNA is the genetic material in the cell, and it provides the code for amino acid sequences. So what we're talking about is protein synthesis. We've talked a little bit about that replication by base pairing, and we'll talk a little more about replication itself, and the critical role of DNA polymerase, which is the enzyme responsible for forming new copies of DNA using a template. As we take a look at this molecule of DNA as it replicates, let's talk about how that replication actually occurs. So as you can see here, every molecule has 2 long chains of atoms that are arranged in a double helix, and there's your double helix. 2 sides or chains have to unwind. And they do this under the influence of the helicose enzyme. And as they unwind, they then separate. In replication, DNA polymerase synthesizes the new strands. And it assures that the nucleotides join with the nitrogen bases. So the nucleotides pair to form 2 new DNA strands. And again, adenine joins with thymine, cytosine joins with guanine. This is the complementary base pairing. And as you can see here, the 2 new strands are identical. The 2 strands are also identical to their parent or the original DNA helix. And don't you love the fact that they call them the daughter molecules and not the son molecules? Sorry guys, but I like that. And that's just the explanation of what we just talked about. So if you look at it, then you can see the adenine and thymine, and you can also see the guanine and the cytosine. Now let's turn our attention to RNA. RNA, like DNA, has a nitrogen base, a sugar, and a phosphate. However, the nitrogen base has a difference. It has uracil as a pyrimidine base instead of thymine. So that's an important difference in this structure, in this nitrogen base pair in RNA as opposed to DNA. You still have nitrogen base pairs, but uracil is present instead of thymine in the RNA molecule. Unlike DNA, RNA is a single-stranded structure. Now, RNA, ribonucleic acid, is responsible for transcription and translation. So it carries out the instructions of the DNA for protein synthesis. In transcription, there is the formation of messenger RNA, and this takes, again, an enzyme, RNA polymerase, for synthesis. And it also has to have binding sites. In that translation, then, you have the synthesis of the polypeptide by using the transfer via that transfer RNA. Now this is an important thing also about RNA. There are three types of RNA. There's messenger RNA, ribosomal RNA, and transfer RNA. And it's important that you understand the function and how they work. Because when you think about DNA and RNA, if you alter any one of these steps or structures, that would then lead to a mutation, and that then leads to a genetic alteration that may or may not be significant. So let's take a look at that structure. Again, you can see the single-stranded structure. It looks very similar, but it is different because of only the single strand. You can see the sugar and the phosphate as part of that, and then your nitrogen base pairs attach onto that. And you can see your adenine, uracil, guanine, and cytosine, which are your nitrogen base pairs. Let's talk then more about these different types of RNA. Messenger RNA is the first type of RNA. The messenger RNA makes up about 5% of the RNA in the cell. And this messenger RNA will vary in size as well as its base sequence. But remember, this messenger RNA has to carry information from DNA to the ribosome. So then we go to the ribosome. The ribosomal DNA has complexes that read the messenger RNA and then helps translate that information into proteins along with the transfer RNA. So you have messenger RNA going to ribosomal RNA, which works with transfer RNA in order to have that genetic message. Now, the transfer RNA carries proteins to match the messenger RNA code. So as a result, then, the ribosomes and the transfer RNA are working together to actually read that messenger RNA-based sequence. I shouldn't say a sequence, those messenger RNA-based sequences. So see messenger RNA essentially as the recipe, because it copies a section of DNA. That copy then leaves the nucleus and enters the cytoplasm, where the ribosomal RNA and transfer DNA then work together to read those base sequences and help build new proteins. It is just such an elegant and beautiful system. Chromosomes, then, are those long strands of DNA that actually carry our genetic information. We know that in humans we have 23 pairs of chromosomes. 22 pairs are autosomes, and 1 pair is your sex chromosomes. Autosomes are somatic, and these are diploid, and they occur in pairs. Sex chromosomes are a little different. They are haploid, so that you have 1 member of each pair. So as a result there, when the sperm and the ovum meet, it then would make a diploid cell. It's important to remember that genes are found on the same position of the same chromosome. And that's the key to understanding how we were able to then map and understand the genetic sequences. That's why we can map that genome. We also want to remember that since we can then look for those genes in those same positions. Structural changes in the chromosomes may or may not result in disease because some genetic mutations, genetic changes, or changes in a chromosome may not really matter that much. But the more common changes that we see have to do with deletion, duplication, inversion, and translocation. Those are some terms that I expect you to have an understanding of. So then when we think about genetic mutations and we think in pathology, we think about changes in the DNA. A mutation is a permanent structural alteration of the DNA. Frequently this happens in base pair substitutions, or it could be an insertion or deletion of a base pair. So those are that's one of the things we look for. Also, the entire chromosome could be developed, or simply or small parts of the chromosome may be lost, duplicated, or arranged inappropriately or misarranged. When we look more about some of our genetic terminology, penetrance means that we're looking at the proportion of individuals with a mutation-causing disorder who actually exhibit the manifestations of that disorder. So in order for us to see a genetic mutation that causes disease, there had to be genetic penetrance. When we think about the probability that a family member may actually have a genetic-based disease, we're looking at the risk of recurrence. And that's when we talk about, that's how we use that Punnett square to look at probabilities. Because remember, risk is all based on probability. And the risk of recurrence or occurrence, if you want to look at it that way, but it's the probability that the family member will have a genetic-based disease occurs with each pregnancy. And that's something that as we teach parents and families about a genetic disorder, it's important that they understand that it's with each pregnancy. So when a child is born with a mutation, we first look at the parental DNA or the makeup of their chromosomes. If the parental unit, the parental DNA or chromosomal material is not mutated or abnormal, then the child has a new mutation, and frequently this may happen during cell division. Therefore, the chance of having a child with that same mutation would not be common, because remember, you have to transmit this genetic mutation. If the child had a new mutation, didn't come from the parents, then the risk of having another child with that same mutation would be fairly low. However, the child has the ability, because their genetic material underwent a mutation, can pass that mutation on to the next generation. So as a result, what we've been talking about for the most part is the inheritance of mutation. But we also know that mutations can be acquired. We can acquire a mutation through ultraviolet radiation, through chemicals and chemical injury, and even through viral injury. Let's look at some patterns of inheritance and some of those important concepts as we think about genetic mutations that are passed through families that cause disease. So we think primarily about autosomal dominant, autosomal recessive, and the idea of a gender-linked trait. So if someone has an autosomal dominant inheritance pattern, it takes a single gene, a single dominant gene for those effects to be observable. The copy causes the trait to appear. In an autosomal recessive disorder, you need 2 copies of that gene in order for the trait for that trait to appear and have the disease. Now, in general, autosomal dominant and autosomal recessive disorders are actually fairly rare. However, we do know that they do occur. So when we look at autosomal dominant diseases, that we will actually be talking about in other disease states, include polycystic kidney disease and Huntington's chorea. Both of these are autosomal dominant disorders, and they actually also are different from some of the other autosomal disorders in that the penetrance, they have good penetrance, but both of these diseases don't manifest at birth. They manifest during adulthood. Autosomal recessive diseases include cystic fibrosis and one that we've already talked about, sickle cell anemia. So you have to have 2 copies of that recessive gene in order for the trait to appear. We also know that there are gender-linked disorders. And in a gender-linked disorder, the trait is linked to either the X or the Y chromosome. Now, most gender-linked genetically inherited disorders are X-linked because there are few genes on the Y chromosome. So, and in addition to that, most are X-linked recessive genes in which the gene is present in the female but affects the male. So what happens in this X-linked recessive disorder in which the gene is present in the female and affects the male is that the male has the disease-carrying recessive gene on his X chromosome. But he develops the disease because there isn't any balancing or normal allele to counteract those effects. So his X chromosome carries this disease-causing recessive gene on his X chromosome, but because he has a Y chromosome that doesn't have another balancing normal allele, which the female would have, the male has the disorder. He also has the ability to transmit this gene to his daughters. He can't transmit the genes to his sons because it's X-linked, correct. So let's look at a couple of disease states in which we suspect some patterns of inheritance, even though we don't have an individual gene that says, hey, this is where it is. We have it on some of our chromosomal information. In diabetes, when we look at type 1 and type 2, there is some genetic material that we're interested in. In type 1 diabetes, there is some influence of genetics, but we don't know how strong that influence is. So in general, when you look at someone with type 1 diabetes, you don't see that necessarily tracking through families. This is very different from type 2 diabetes. In type 2 diabetes, there tends to be a fairly strong family history, therefore there is more genetic influence. This is especially apparent that there is a link in African Americans. Now, what's interesting about the genetic influence in type 2 diabetes is the influence is in insulin sensitivity and insulin resistance. Does not seem to be associated with beta cells basically wearing out or beta cell dysfunction. It's in the peripheral insulin resistance or sensitivity. We also see some evidence for transmission of type 2 diabetes for age, onset, as well as prevalence. Now, another very interesting thing about diabetes is the presence of suspected suppressor genes. And this occurs in diabetic nephropathy. You knew, once again, I'd get it down to kidneys, didn't you? But there is an evidence of predisposition related to suppressor genes in diabetic nephropathy. So what we're looking for here is the role in both protection from as well as the occurrence of diabetic nephropathy in our genetic material. Another disease state where there's been a lot of work is in high blood pressure. And the genetic piece they're looking for there is salt sensitivity, sodium sensitivity, so that there may be a genetic predisposition for an individual to have sodium sensitivity, which then affects their blood pressure control. But not everyone has this. That's why not everyone who has high blood pressure is sensitive to sodium. Last but not least, depression. they've identified that depression is 1.5 to 3 times more likely in an individual who has a first-degree relative who has depression. So how do we sort this out? Well, that's where a Punnett square comes in. A Punnett square, and your textbook goes through this as well, is a visual method to determine the probability of an offspring having a known genotype. And you have to have a known genotype for this to work. So that's how you use that autosomal and recessive genetic information. And again, the probability, because this is all about probability, applies to each pregnancy because that's where you have the mixing of the genetic material. Each pregnancy stands alone in terms of probability of risk. So when you look at a Punnett square, the capital letters indicate the dominant trait. Let me go to one. So the capital letters indicate a dominant trait. The lowercase letters indicate the recessive trait. So small h, big H, uppercase. So this would be dominant. This would be recessive. We also generally the way we use these is the male is across the top, the female is across the side. Oh, and I says gene along the right side. I'm so sorry. That's the left side. I will make sure that is changed. So let's look at a normal homozygote, which means they have the same kind of genetic, both genes are the same, and one heterozygote individual with a dominant disease. So as you can see, you just cross them, H to H. This H goes with this H. Small h, big H, and 2 small h's here. So as a result, then, there's a 50% chance that their offspring would have the disease. There's also a 50% chance that there is no altered genetic material. Let's look at another example, and this would be a gender-linked recessive disorder. So if you look at the gender linking, here's your gender, here's your X and your Y. Again, you use your same pattern. You have a 50% chance that the male offspring will have the disease. So here's your disease causing, or actually since it's recessive, here's your disease causing, and here is your normal. So there's a 50% chance that the male offspring has the disease and a 50% chance that passes the trait to a daughter. A couple of other things that I think are worth talking about, even though this isn't a pharmacology course, and I realize that, but thinking about how pharmacology plays with pathophysiology can also be very important. One of the most common areas that we've looked at is the cytochrome P450 enzyme system. This is a group of at least, I do mean at least because they continue to identify additional proteins, but there are at least 57 different proteins, each coded by a different gene. And of course, these proteins are enzymes. It's estimated that about half the population actually have genetic variations in this cytochrome P450, the CYP450, that may affect drug metabolism. And if you affect drug metabolism, that then alters the safety and potentially efficacy of the drug. And the reason I put this in pathophysiology is because of the safety issues that could lead to pathology. It's estimated that there are 3 main enzymes in this cytochrome system, and they're responsible for about 45% of the drug metabolism. So what this results in then, in this genetic makeup, is that some individuals are poor metabolizers and other individuals are rapid metabolizers. If an individual is a poor metabolizer, they lack the functional enzymes for metabolism. As a result, then, they don't get as positive effect. They have less effect from the medicine. medication. If they are a rapid metabolizer, they have a higher rate of metabolism through this enzyme system. As a result, then, they have a greater risk of adverse drug reactions, and adverse drug reactions can lead to pathophysiology or a pathologic response. Now, not all drugs are metabolized by the cytochrome P450 system, but when you get to pharmacology, you'll talk about this again because it's important to know which ones are metabolized by this system, and sometimes we need to investigate that. A great example of some of the investigation that has gone on with this is, once again, in depression and looking at antidepressants. They've actually been able to identify 3 areas of known polymorphisms in this system. In the psychiatric population, there's about a 57% risk that they have an altered cytochrome P450 genetic alteration, as opposed to or compared to about 35% of the genetic population. And the way they're using this in psych is if they are giving a medication and they're just not getting the therapeutic effect, they will check their cytochrome P450 material to see whether or not they have those altered genes that are affecting their drug metabolism. Pharmacogenetics is a huge and rapidly expanding area in our practice. So what about genetic testing in general? I think it's important. I think this isn't pure patho, and I recognize this, but I think it's we need to understand the availability and the ease of availability in genetic testing. Proprietary labs offer genetic testing. I mean, all of those ads that you see on how to know what your background is and you thought you were Italian but you ended up being Scandinavian. Same idea. The cost varies a great deal and results can come back within a few weeks to several months. But that puts your genetic material out there. So some of the examples of genetic testing that we've been doing for a number of years include cystic fibrosis, the BRCA1 and 2 genes, which tells us about the risk for developing breast cancer, as well as potentially prostate cancer. In kidney world, we look at polycystic kidney disease and Alport system. Polycystic kidney disease manifests itself in early adulthood. Alport system manifests itself usually by the age during early childhood. We talked about type 2 diabetes and cytochrome P450. The other piece is this issue of newborn screening. In Texas, we screen for 55 disorders at birth. This is part of the expectation, and we are mandated to do this by law. So 55 genetic disorders are screened in Texas. The first genetic screening is done in the first 24 to 48 hours after birth, and then the second screen is done 7 to 14 days following that to look for penetrance to make sure that we've caught those genetic influences of those that we can test for. So we've talked about how genetics can influence both the incidence and the appearance as well as potentially progression of diseases. Genetics influence how we respond to therapeutic interventions, that whole idea of pharmacogenetics. But it's important that we understand those concepts of genetics and that understanding increases our ability to both recognize, treat, and evaluate treatment of many of our disease states. It also helps us help patients as well as parents who are our patients understand disease states that their children or their family members may also have. You will continue to use this information, and I hope this information will really help you when you do your genetics paper, where you'll have the opportunity to delve into more completely describe a genetic disorder in which you are interested. If you have any questions, please let me know. And as always, thank you for your attention.